The human gut is home to trillions of microorganisms that form a complex ecosystem we call the gut microbiome. And among the many molecules these tiny inhabitants produce, one stands out for its fascinating dual nature as both a potential toxin and a crucial signaling molecule. Hydrogen sulfide, that infamous gas with the smell of rotten eggs, has long been known as a dangerous substance in industrial settings. But science has revealed something remarkable about this simple molecule composed of just two hydrogen atoms and one sulfur atom. When produced in the right amounts and in the right places within our bodies, hydrogen sulfide acts as a sophisticated signaling molecule that influences everything from blood vessel function to nerve cell communication. And while our own cells can produce this gas through various enzymatic pathways, there's another source that's been hiding in plain sight all along, living within the twisting corridors of our intestines. The bacteria that call our gut home are prolific producers of hydrogen sulfide. And as researchers have delved deeper into understanding the microbiome, they've discovered that this bacteria-derived hydrogen sulfide plays a pivotal role in maintaining health and contributing to disease in ways that are only now beginning to be fully appreciated. The story of hydrogen sulfide in the gut is intimately connected to the broader narrative of how our microbiome communicates with the rest of our body, a concept that has revolutionized our understanding of human health and has given rise to the study of various biological axes that connect distant organs and systems through the mediating influence of our gut bacteria. The concept of the brain-gut axis was among the first of these paradigms to capture scientific attention, representing the original framework through which researchers began to understand that the gut and its microbial inhabitants don't exist in isolation, but rather engage in constant bidirectional communication with the central nervous system. This revolutionary insight opened the floodgates to discovering numerous other axes, each representing a unique pathway of communication between the gut microbiome and different organ systems throughout the body. Today we recognize the gut immune axis, which explains how intestinal bacteria shape and modulate our immune responses, the gut-heart axis, which reveals the profound influence of gut health on cardiovascular function, and the gut-endocrine axis, which demonstrates how microbes can affect hormone production and metabolic regulation. What ties all these axes together is a common theme of microbial composition changes, alterations in intestinal barrier function, and inflammatory responses, and hydrogen sulfide emerges as a key player in modulating many of these processes. When the delicate balance of the gut microbiome is disrupted, a condition known as dysbiosis, the production of hydrogen sulfide can swing dramatically in either direction, with concentrations becoming either too high or too low, and these imbalances ripple outward through the various axes to contribute to disease development in multiple organ systems. Understanding bacteria-derived hydrogen sulfide, therefore, becomes crucial not just for appreciating gut health in isolation, but for comprehending how disturbances in the intestinal environment can have far-reaching consequences throughout the entire body. The intestinal barrier represents the first line of defense in these complex interactions, serving as both a protective wall and a selective gateway that must perform the delicate balancing act of allowing nutrient absorption while preventing harmful substances and pathogens from entering systemic circulation. This barrier consists of a single layer of epithelial cells held together by tight junction proteins, overlaid by a mucus layer that serves as both physical barrier and habitat for commensal bacteria. Hydrogen sulfide produced by gut bacteria has been shown to have significant effects on this barrier function, though these effects can be beneficial or detrimental depending on concentration and context. At physiological levels, hydrogen sulfide can promote barrier integrity by enhancing tight junction protein expression and supporting the production of protective mucus. But when dysbiosis leads to excessive hydrogen sulfide production, particularly from sulfate-reducing bacteria that can proliferate under certain dietary and disease conditions, the gas can become toxic to colonocytes, the cells that line the colon. This toxicity can lead to increased intestinal permeability, often referred to as leaky gut, where the barrier becomes compromised and allows passage of bacterial products, undigested food particles, and other potentially harmful substances into the bloodstream. The inflammatory response that follows such barrier disruption is a common thread connecting dysbiosis to systemic disease, and hydrogen sulfide's role in either maintaining or compromising barrier function makes it a critical factor in determining whether the gut remains a sealed ecosystem or becomes a source of inflammatory triggers. In the context of intestinal inflammation, such as that seen in inflammatory bowel diseases like Crohn's disease and ulcerative colitis, the relationship between hydrogen sulfide and mucosal healing becomes even more complex, as the gas has been implicated in both perpetuating inflammation through barrier disruption and potentially supporting healing processes through its signaling functions, suggesting that the key to therapeutic manipulation may lie in achieving optimal rather than maximal or minimal hydrogen sulfide levels. The influence of bacteria-derived hydrogen sulfide extends dramatically beyond the gut itself, reaching into the realm of neurological health through mechanisms that are as intriguing as they are concerning. Alpha-synuclein is a protein that has gained notoriety for its role in Parkinson's disease, where it misfolds and aggregates into toxic clumps that accumulate in brain cells, leading to the characteristic motor symptoms and neurodegeneration associated with this condition. Recent research has revealed that the gut microbiome and its metabolites may influence alpha-synuclein aggregation, and hydrogen sulfide has emerged as a potential contributor to this process, with studies suggesting that excessive hydrogen sulfide can promote the misfolding and aggregation of this critical protein. The brain-gut axis provides a physical and chemical highway through which signals from the gut can reach the brain, including the vagus nerve, which directly connects the gut to brainstem regions, and metabolites like hydrogen sulfide that can enter circulation and potentially cross the blood-brain barrier or influence brain function through indirect pathways. This connection has profound implications for understanding neurodegenerative diseases, as it suggests that managing gut health and bacterial hydrogen sulfide production might offer novel therapeutic strategies for conditions traditionally viewed as purely neurological. The gut-heart axis reveals another dimension of hydrogen sulfide's systemic influence, particularly in the context of ischemic injury, where tissues are deprived of oxygen-rich blood. Hydrogen sulfide has well-documented effects on the cardiovascular system, and while endogenously produced hydrogen sulfide has been extensively studied for its cardioprotective properties, including vasodilation and protection against ischemia-reperfusion injury, the specific contribution of bacteria-derived hydrogen sulfide to cardiovascular health and disease remains an area of active investigation. In ischemic injury, whether in the heart during a heart attack or in the brain during a stroke, the balance of hydrogen sulfide becomes critical, as appropriate levels may support cellular survival and recovery, while excessive levels could contribute to tissue damage and dysfunction. The gut-endocrine axis brings metabolic syndrome into focus, that cluster of conditions including hypertension, atherosclerosis, insulin resistance, and obesity that collectively increase the risk of cardiovascular disease and diabetes. Hydrogen sulfide's role in blood pressure regulation is multifaceted, as the gas can act as a vasodilator, relaxing blood vessel walls and lowering blood pressure. Yet dysbiosis-associated changes in hydrogen sulfide production may contribute to hypertension through complex mechanisms involving inflammation, oxidative stress, and alterations in other vasoactive pathways. Atherosclerosis, the buildup of plaques in arterial walls that underlies most heart attacks and strokes, has also been linked to gut microbiome composition and metabolites, with hydrogen sulfide potentially playing both protective and harmful roles depending on the context. Some studies suggest that hydrogen sulfide can protect against atherosclerosis by reducing inflammation and oxidative stress in blood vessel walls, while others indicate that excessive levels or production in the wrong locations might contribute to plaque formation and instability. The connection to glucose regulation and metabolic health adds another layer to this complex picture, particularly through the influence of hydrogen sulfide on glucagon-like peptide 1, or GLP-1, a hormone that plays crucial roles in insulin secretion, glucose homeostasis, and appetite regulation. The gut microbiome is known to influence GLP-1 production through various mechanisms, and emerging evidence suggests that hydrogen sulfide may be one of the signaling molecules through which bacteria communicate with neuroendocrine cells that produce this important hormone. This connection is particularly relevant given the current therapeutic focus on GLP-1 agonists for treating diabetes and obesity, as it suggests that manipulating gut bacteria and their hydrogen sulfide production might offer a complementary or alternative approach to enhancing GLP-1 activity. The gut immune axis weaves through all these other axes, as immune function both influences and is influenced by microbiome composition, barrier integrity, and systemic inflammation. Hydrogen sulfide has been shown to have immunomodulatory properties, affecting the function of various immune cells and the production of inflammatory mediators, and bacteria-derived hydrogen sulfide may therefore play a role in determining whether the immune system maintains tolerance to commensal bacteria and dietary antigens or becomes hyperactivated and drives inflammatory diseases.